Good morning, students. Today we are going to start with a new topic in the e-lectures. It is evolution of sporophyte in bryophyte. So, what sporophyte is basically is? We know the sporophyte is a diploid phase in the life cycle of the bryophytes, and it is very crucial for understanding the reproductive stages. This sporophyte in bryophytes mostly depends on the gametophyte for food and nutrition. So, because of this, we are trying to find out how the sporophyte has been evolved in bryophytes. We know that for this, two theories have been given. One is known as the Antithetic Theory, or it is also known as the Theory of Sterilization, and the other is known as the Homologous Theory, or it is also known as the Reduction Theory or the Retrogression Theory. So, this Antithetic Theory, this was proposed by Tsiolkovsky in 1874, and according to this theory, which is also known as the theory of sterilization, in this the sporophyte is evolved from the simple to the complex form. So this theory was also supported by Bowers in 1890, Strasburger in 1894, and Campbell in 1940. So, according to this theory, there is a progressive sterilization of the sporogenous tissues, and there is a primitive sporophyte like Riccia are simple and fully sporogenous, while the Funaria is highly more complex. So, so when you see this theory, you can see in the Antithetic theory or theory of sterilization, the Riccia, which is having a single jacket layer with all the spores within that being totally what you call the fertile. And this entire, do not have the foot and the seta, and it is being covered by, it is within the gametophyte. When you see in the second stage, when I say that further sterility takes place in case of the Marchantia, there is foot and seta, and the capsule is a single jacket layer, and in the spores there are certain elaters, sterile structures called elaters. Well, in Palea, further sterility of the sporogenous tissues takes place, and the sterile tissues further develops. The elaters, the pseudoelaters, and multi-jacket capsules are based. So, anthroceros, there is a thick columella comes between that and that one. Further progression, you can see the sterilization takes place in Funaria, where you see the counter, the within is there is a spores, and that the multi-layer jacket cell is there, foot is there, outer wall consists of the photosynthetic tissues, function, photosynthetic cells are present in this. There are so the most of the sterilization is because of the perform various types of the function where the outer cell will helps, the foot will helps in the anchorages, theta helping the stalk supporting the capsule, and also the photosynthetic cell when present, they helps in the process of preparation of the for food. So this is about the antithetic theory or the theory of sterilization, right from Riccia to the Funaria. There is continuous sterilization of the sporophytic. There is another theory that is what we call it as the homologous theory, or it is also known as the retrogression theory or retrogression sterilization of sporogenous tissue or reduction theory. This was proposed by Princeton in 1876. Sporophytes are modifications of the gametophyte, where the ancestral bryophytes had an independent sporophyte. So the evolution leads to the reduction in the sporophyte complexity. When you see this, this is the homologous theory or a theory of sterilization. Here, you can see from the Funaria, which is more complex, where the sterile cells are more in the capsule. It is from Anthroceros that has been reduced from multi-jacket to single jacket, and here from Palea again there is still elaters are there, but still multi-jacket structures are there. Marchantia, the capsule is only single jacket, foot and seta are present, but the foot and seta here the stalk is not very long. And when it comes to the Riccia, the particle completely foot and seta are completely reduced it off, and only the all the sporogenous tissues that are present within the capsule are all fertile. And this theory was also supported by Church, Zimmermann, Fritsch, and Wood. So evolution has been traced through various stages from simple forms to Riccia to more complex forms seen in Funaria. Each stage demonstrates progressive changes in the structure, function, highlighting the evolutionary adaptations of the bryophyte. So according to homologous theory, the sporophyte and the gametophyte generations are fundamentally similar in nature, and the sporophyte is the direct modification of the gametophyte and is not a new structure or type. So this is the structure. So you can see the Riccia capsule where within the archegonia, the venter is the spore mother cell, the zygote developing the spore mother cells and our cells and developing the capsule. While in case of when you see Marchantia, there is a foot, seta and the capsule having the elaters with the calyptra is there. So these are the further sterilization. Further sterilization in Anthocerates, where you can see the foot embedded within the gametophyte, the meristematic tissues are present, the spore mother cells are present, the spore tetrads are there, along with the spore tetrads there are elaters and pseudo-elaters. Further modifications, the foot, seta and capsule, they were little bit independent life. They have a trichoblast-like structure, the peristomial teacher, the operculum is there, and within the spores are highly reduced and they are present in the sporangial walls. And within centrally is present what you columella is present, sterile area columella. So, right from the Riccia up to the Funaria, according to Antithetic Theory, the progressive sterilization takes place, and homologous theory from Funaria to Riccia, there is a retrogression of the sterile structures and further fertility will be developed. So, so this is Bryophytes, the simplest to the more complex form. So first is simplest is Riccia, second is Marchantia, and the third is Funaria. And in all, we can say that the evolution of sporophyte illustrates the plant adaptation, and the theories provides insight into the sporophyte development, and understanding this concept is key for any of the biological student. Thank you for understanding the evolution of the sporophyte, showing the two theories how the sporophyte has been evolved.